Hello,
Yes, sir.
hello.
Yes, sir. Good evening, sir.
Is it audible to you?
Yes, sir.
Hello,
Yes, sir.
hello,
Hello. Yes, sir.
hello.
Hello. Yes, sir.
Good evening, all.
Good evening, sir.
So, right, yesterday we started about pollinology. In pollinology, that is the study of pollen grains. In this, we discussed about first of all pollen morphology. The pollen morphology necessary to plant identification at taxonomic point of view. It is very important. Let's see the pollen morphology. Uh, Let me share PPT. BRL images files. No, that's not in gallery. Just wait. File compressor, extract compressor, convert PDF. The small problem, just wait. Right. Are you able to see PPT slide? Hello? Hello?
No, sir. No, sir. Yes, sir.
Hello?
Yes, sir. Yes, sir. It is visible.
So in pollen morphology, there are few the basic units such as pollen units. That is, they are very important in taxonomic view. First of all, pollen units. Second one, polarity, symmetry, shape, size, and aperture. So these are the morphological characters. The pollen grains
Sir,
have been categorized into different groups.
sir, your
First, let's
voice is
see the
not
pollen
audible.
unit. Pollen unit means the pollen grains are produced within the anther. You know, already we discussed in first unit about the microsporogenesis. What is
Excuse me, sir.
microsporogenesis?
Excuse me, sir.
Microsporogenesis means production of microspores in anthers. which are developed by meiotic division. Right after what happened after meiosis division in microspore mother cell, actually the pollen grains appeared in tetrad condition. The tetrad may be isohedral, bilateral, linear, different type. Already we have seen that. So, but when it is maturity, they are separated into unique one. That means separated into one. The most probably the most of the plants, pollen grains, they are after development they are separated into individually. So the type of pollen grains called as monad. So based on unit, the pollen grains actually do not remain united at maturity stage, and they are disassociated into single pollen grain. If the pollen grains in single, that is called monad. Sometimes rarer types like dyad, that means two pollen grains together attached. So that condition called as dyad. Octant, 8 pollen grains and more days that considered as a polyad condition. So 90% of the non-pollen tags
Excuse
are
me,
the
sir.
monad condition. But very rare condition we can observe polyad condition. In polyad condition, the best example acacia pollen. In acacia
Yes,
pollen,
sir.
total 16 pollen grains attached. So
Sir, excuse me,
that is
sir.
called polyad condition. So based on pollen unit, we can classify the pollen grains into monad and dyad and polyad. So that is number one. And
Sir, excuse me, sir. Sir, sir.
we can see in the monad polyad, they are all polyad types. Second morphological is
Excuse me, sir.
most important criteria and identification and giving description to plant identification. As aperture position in this polarity recognition, we take consideration the aperture position. As the aperture position is of primary phylogenetic and functional significance. In that, actually all pollen grains are tetrad stage during development, and the polarity is determined in this stage prior to the separation. So we can identify it only the polarity when they are tetrad condition. But after it is separated, we can't identify the polarity. Here, the part of the pollen grains which is nearest to the center of is the proximal pole, and toward the opposite called distal pole. Let me draw simple. Pollen grain. The part of the pollen nearest to the center. This is the center actually. Uh,
Sir,
In the tetrad, the
sir,
proximal
excuse me,
pole
sir.
and the toward the opposite side distal pole. That means if the pore is present here, yellow color,
Sir, excuse me,
that
sir.
is proximal pole, and exact opposite, this is distal pole. This is distal pole. The center nearest to the center, this one proximal, and this one distal pole. So the imaginary line between the proximal and distal pole, that is called polar axis. This line, let me draw this line, this imaginary line between polar axis, proximal and distal pole. The imaginary line called as polar axis. and perpendicular to the another line, the polar axis, that is called equatorial axis. This is the color, uh, the blue color, equatorial axis, yellow color, which connected the proximal pole and distal pole. The both poles, proximal, distal, yellow color, that is considered as a polar axis, PA. This one is polar axis, PA. So that means
Sir,
if the
excuse me,
both are
sir.
same line, that means the proximal and distal pole same line, that is called
Sir,
isopolar.
excuse me, sir.
That condition called isopolar. If they are different line, that is called as a heteropolar. Most of the pollen grains are isopolar. So the polarity also important characters to identify the pollen grains. And next one, after polarity, another important character is symmetry. First one has to completed, that is pollen unit. Second one is polarity. In polarity, we can classify it into isopolar if the same lane, the proximal and distal pole on the same lane, that is called isopolar. If they are different, their colors heteropolar. Right. And next third aspect is, third one is symmetry type. Let's say symmetry.
Hello, sir. Excuse me, sir.
Polarity completed. And where is symmetry? Okay.
Sir, excuse me, sir.
Here given actually in polarity, the pollen grain may be either apolar, polar. In apolar spores and poles, polar region cannot be distinguished because we cannot find out where the proximal and distal. If you see here, so in isopolar condition, proximal, distal pole, same line. Here proximal, sorry, distal pole is present. In heteropolar condition, where is proximal pole? In proximal, no pole present. So this condition called as heteropolar, right? And next one. And third aspect is symmetry. First pollen unit complete, polarity complete, and third aspect morphologically study symmetry. Symmetry grains or spores are asymmetric. Let's discuss about asymmetric condition. The asymmetric grains either non-fixiform. That means actually asymmetric means there is no proper shape. If there is no proper shape to any pollen grain or spore, that is considered as a asymmetric. In asymmetrical grain, have no plane of symmetry. No plane of symmetry. They are rare in grains. Symmetric cases, most probably we can identify most of the plant taxa. Again, the symmetric to symmetry and bilateral. Already really means we can cut any plane. It gives two equal parts, any direction. That is radial. Bilateral in the name given, the bi means if you cut one plane which divides two equal parts, that is called bilateral symmetry. So let's see again here. The radial symmetry and bilateral having single plane of symmetry. Only single plane. If you cut the single plane, then only give the equal parts. That is bilateral. In diagram,
Excuse me, sir.
given a letter, for example, symmetry, sorry, Suhana,
Yes, sir.
hello,
Hello, sir. Sir, sir, am I audible?
hello,
Sir, am I audible, sir? Sir,
Suhana.
sir,
Hello,
hello, sir. Sir, am I audible, sir? Sir, sir,
hello,
hello, sir. Hello, sir. Hello, sir.
hello,
Hello, sir.
hello.
Hello, sir. Hello.
Hello, participate to
Yes, sir.
unmute themselves.
Hello, sir. Am I audible?
Suhana,
Yes, sir. Hello, sir.
unmute, unmute. You do unmute,
Yes, sir. I unmuted.
but your voice not audible. When I see the speaker, but is it is seems to be appear as a you are trying to talk.
Sir, am I audible now? Sir, 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 am I audible now? Sir, hello, sir, am I audible now? Sir, am I audible now?
Hello?
Sir, am I audible?
Hello?
Hello, sir. Hello, sir. Sir, am I audible? Sir, sir.
Hello?
Yes, sir. Am I audible?
Now clear.
Sir, I'm speaking, sir.
Just wait. Now audible to you?
Yes, sir.
Hello?
Hello, sir. Hello, sir.
Here. Now
No, sir.
my voice audible.
Yes, sir. Little bit.
My voice is clear or not?
Yes, sir. Now it's clear. Sir.
Hello?
Hello, sir.
Hello?
Hello, sir. Sir, I think it's network problem, sir. Sir.
My voice
Sir, sir, it is not clear, sir.
now clear?
Yes, sir.
Your voice clear to me. Your voice audible to me.
సార్ మీ వాయిస్ వినిపించట్లేదు అప్పటి నుండి. నవ్ ఇట్స్ క్లియర్ సార్, ఇప్పుడు వినిపిస్తుంది.
Clear?
ఎస్ సార్.
Right. So far what we discussed, you did not see anything, right?
ఎస్ సార్. సార్ ఐ సీ ఓన్లీ ద pollen units ఓన్లీ.
Only pollen unit visible and audible both.
ఎస్ సార్.
Right.
ఫ్రమ్ pollen
After
unit
pollen,
it is disturbed సార్ ద వీడియో.
okay. After pollen unit, polarity, you did not listen,
ఎస్ సార్.
and symmetry also.
నో సార్, సిమెట్రీ అండ్ polarity కంట్రోల్ సంథింగ్.
Okay, university host actually the given all are mute. That's why it is inconvenience to us. So anyway, I will explain from the polarity.
ఓకే సార్.
Pollinate, I think already. Should I explain pollen unit?
ఎస్ సార్, వన్స్ అగైన్.
Pollen unit simple. Pollen unit, that is number of pollen grains. You know already microsporogenesis. What is
ఎస్.
microsporogenesis?
ద ఫార్మేషన్ ఆఫ్ మైక్రో pollens.
Right, formation of microspores. Actually, after formation of microspore, these microspores are pollen grain in the tetrad condition. But when reach maturity, they are individually separated. Mostly, they are separated into single. So the single pollen grain called as monad. But very rare cases, they are unite as a two. That is called dyad. And another rare cases, they are more than two. That is called as a polyad condition. This is called as pollen unit. Is it okay?
ఓకే సార్.
Right. And next one, polarity. Let me share PPT. Oh no. In polarity. the simple polarity based on the proximal pole and distal pole. Are you able to see the polarity?
Yes, sir.
Right. Polarity happen. Mostly polarity we can identify it when in tetrad condition means 1, 3, 4. 4 pollen grains. This is called tetrad condition. In tetrad condition, this is the center C for center. And what the center pole? This is called proximal pole. Opposite distal pole. So imagine a line this between proximal and distal pole. This is called as a this imaginary line called as a polar axis. Two poles. Two poles imaginary line polar axis, which is called as polar axis shortcut PA, and another line imaginary line perpendicular to the polar axis. This is called as equatorial axis. Equatorial axis, right? So in reality, there are mostly three types. Apolar, there is no in particular shape, that is called apolar, and isopolar and heteropolar. In actually in apolar, there is no aperture, there is no pore. In isopolar, the polar axis, proximal pole and distal both are same line. If the both are same line, that is called considered as the isopolar. If only one pole present, either proximal pole or distal pole present, that is called heteropolar. Only one is present, another not present. That is called heteropolar. And we can see the diagram also. Just wait. If we in this is apolar, first one is apolar. Apolar, there is no proximal and distal pole. That is called apolar. In heteropolar, if you see here, this is proximal pole, sorry, distal pole. In distal pole, one aperture mark is present. But where is this distal pole? Where is proximal pole? Proximal pole, no pore. If you see isopolar, proximal, sorry, distal and proximal. Proximal, remember, always toward the center. So if the both marks are present, that means distal and proximal. That is called isopolar condition. Is it clear? Hello?
Yes, sir.
Polarity clear? Right. Now after polarity, another important character symmetry. Let's say let me take a PPT slide. So now third aspect is symmetry. In symmetry, pollen grains or spores are symmetric. Actually, there are three types: asymmetric. In symmetric, two types. One is radially symmetry. Second one is bilateral symmetry. Asymmetric, first one. Asymmetric one. And in symmetric pollen grain, two types. One is radially symmetry and bilateral symmetry. In asymmetric, no proper shape. The pollen grain does not have proper shape. That is considered as a, that means we cannot cut any plane. We cannot cut any plane to give equal part. That is called asymmetric. If you see the radial symmetry, name itself given radially. Radially, if you cut any plane, it gives two equal parts. For example, if you see the diagram A, in diagram A, if you cut two equal part, two equal part, two equal part. In the B diagram also same, equal, equal, equal. But in C and D diagram, if you cut this plane, only give the two equal part. If you cut this plane only, sorry, this plane this plane only two equal part. That is called bipolar. Sorry, bilateral symmetry. Is it clear? Symmetry? Hello?
Yes, sir.
Right. The another third aspect is morphological aspect, shape. The shape of the pollen grains varies from species to species. This shape also useful in spore and pollen grain identification. The shape may be taken consideration based on polar axis distance and equatorial axis distance. Let's see how the polar axis and distal polar axis and equatorial axis are involved in to identify the shape. Let's see. Right. Actually, scientist Erdmann 1952 categorized shape process based on the told you. Based on that, we can classify it into a different there the shape of cluster, prolate, spheroidal, subprolate, perprolate, oblate, oblate spheroidal, suboblate, peroblate. These are all. First of all, A diagram, peroblate, B diagram, oblate, C diagram, suboblate, D oblate spheroidal, and next E spheroidal, prolate spheroidal, subprolate, this is prolate and perprolate. So these are the different type of shape. This elongated, this is called prolate shape. Round like this round, that is the spheroidal. And somewhat like this egg-shaped structure, that is called half-blade shape. Is it clear? Shape also clear?
Yes,
Hello?
sir. Yes, sir.
Right. Now size. So the size also taking consideration to plant identification. The pollen grain shows great variety in their size from smallest to largest. Here the smallest pollen grain, the noted in Mysotis, which is present in Boraginaceae. Sorry, right, Boraginaceae family. And largest pollen grain present in, I think, just wait. Largest one, I think Mirabilis ariaceus. Let's see. Are you able to see?
Yes, sir.
Just one. Aperture,
Yes, sir.
largest grain present in Mirabilis. And now another consideration to pollen taxonomy, aperture type. An aperture type, what is aperture? Aperture, the small opening place. Let me draw here. For example, this is pollen grain. The pollen grain have the germ pore. The germ pore, if it is round in shape, that is called porate. Round in shape, that is called porate aperture. If the pollen grain, the aperture like a elongated structure, that is called carpet, pollen. Pore like aperture, that is called porate. If the both are present, if the both are present means if you take the pollen grain, both carpet, pore, both are present, that is called calporate. That is called calporate. And in this aperture also, we can give number of aperture. Only pores three, for example, if this is the pollen grain, in this pollen grain, three pores are present. Three pores. This is called triporate. Tri means three. Triporate. If the four present, tetra. Five present, this is called pentaporate or polyporate condition. And if you take the same way, the carpet also three present tricarpate, more than five, that is called polycarpate. And carporate also same. If it is three number tricarporate, one number monocarporate, two number dicarporate. Like this, that this aperture types taking consideration to identification. And another this among this, the aperture type is most important to our identification of pollen grain. And actually not given here. The another aspect, the seventh one, exine ornamentation. If you take the exine ornamentation, hello,
Yes, sir.
whiteboard visible or not?
Yes, sir.
Right. The another morphological aspect, actually not given here in our slide. Uh, I don't know whether in our book given or not, but remember this is also important. Exine ornamentation. Also, exine ornamentation. If you take the pollen grains, pollen wall to the first intine outer exine, the ornamentation appear in the exine only. We will discuss later the stratification of pollen wall also. I will discuss later tomorrow. I will give you. And just here listen about the exine ornamentation. In exine ornamentation, first of all, silate condition. Silate condition means smooth. If the pollen exine surface without ornamentation, smooth appearance, this is called silate condition. And scabrate. In scabrate, this intine, the exine, the somewhat. Let me take another PPT. So, rough nature, not smooth. When compared with silate, some rough nature, that is called scabbate. And another one is granulate. In granulate, this is pollen wall, and pollen wall, small granules are present like this. Small granules are present. That is called granulate. And echinate. In echinate condition, for example, this is echinate. In echinate condition, the echins are present on the pollen surface like this. Echins also present on the pollen wall. So this condition called as echinate. And another ornamentation, very important one, most probably most of the plant shows this type of ornamentation, that is reticulate type. In reticulate type, on the pollen wall, net is present. Net-like appearance, zigzag, net-like appearance. If the net-like appearance present in exon ornamentation, that is considered as a reticulate type. That is considered as a reticulate type. Is reticulate type. So anyway, pollen morphological study important in essay question. This is very important in essay question. And so again, once again, I will discuss about the pollen morphological study. The why it importance pollen morphological study? Why it is important? Why we study the pollen morphology? What are the pollen morphological aspect? Hello?
Yes, sir.
Is it audible or not?
Yes, sir.
Right. Pollen morphology aspect. There are given 7 or 8 aspects mainly. For these aspects used to based on this morphological character, we can identify the plants. We can identify the plants. In pollen taxonomy, we can identify the plants based on the morphological character. What are the morphological character here? Number one, pollen unit, one of the morphological unit. In pollen unit, maybe these are the mostly pollen grains are when you estalysis method, that means the study of pollen morphology by estalysis method. When we do estalysis method, pollen grains appear in single. That is that is one type of criteria. And if pollen grains are more than one, that is called polyad condition. And second criteria is polarity. The polarity apolar, isopolar, heteropolar. And third one, symmetry. Symmetry, 3 types. What are the 3 types in symmetry? Hello?
Radial symmetry, bilateral symmetry.
Right. Asymmetry first, asymmetry, radial symmetry, and bilateral. After that, after symmetry, shape. Shape. There are, according to Erdmann, there are 8 types of shapes. They are first of all oblate, parablate, oblate spheroidal, and prolate spheroidal, and prolate, periprolate. These are all based on shape. Shape also one of the criteria. After the shape, size. Actually, I forgot that the size measurement taken by polar axis and equatorial axis. You know already polar axis, where the polar axis and where the equatorial axis. Polar axis present between the distal pole and proximal pole, imaginary line. Both are imaginary lines. Right. That size and aperture. How many types of aperture? Hello? What is aperture first?
Opening.
Aperture means
opening.
Small opening. That is germ pore opening. That means from these openings, germ tubes are developed. So how many types of apertures here?
2 types. Monocolporate.
What are those?
Monocolporate and dicolporate.
Only 3 types. No. That's mono and di. That is quantity based on the characteristic nature of the aperture. Actually, these aperture are 3 types. If the pore is round shape, that is called porate. Slit-like elongated structure, that is called colpate. Colpate. If the both are present together, the both means pore and colpus. Both are present, that is called colporate. If the elongated structure present in deepen, that is another one, sulcus also there. But very rarely discover the sulcus. Mostly it is present in the monocot family. But majority we find out porate condition, colpate condition, colporate condition. So that is next. That means triporate, tricolporate. diporate, that is the next one. That is the based on the number of the aperture. But we discuss characteristic nature of aperture first. Based on characteristic nature, actually there are majorly three, total four. The three major types are porate, colpate, colporate. Last one is sulcus, sulcate. Sulcus also present in some monocots pollen grains. And this is aperture types. And the last one, what is one? What is last
Exam,
morphological character?
exam ornamentation.
Exon ornamentation, simple. Exon ornamentation may be salate, may be scabate, may be granular, echinate, or reticulate. That is the based on ornamentation type. So these are the major criteria to identify the pollen morphology to recognize plant taxa. Right? Is it okay? Clear?
Yes, sir. Yes, sir.
Any doubt? Any doubt today class?
No, sir. Sir,
We will
sir,
discuss tomorrow.
sir, when will be the practicals, sir?
Tell me.
Practicals, sir. When will be the practicals?
Practical training classes or practical exam?
Practical classes, sir.
Practical training classes.
Yes, sir.
Now presently MS PG practical classes conducting now. So
Okay.
maybe after practical classes, 6th semester exams, after that 4th semester, maybe next month.
Okay, sir. Okay, sir.
Okay, anyway, we will inform you. Just keep in touch with the website. Do you
Okay,
have
sir.
group?
No,
Do you have
we have friends group, sir.
Okay. And friends, you can let you know about the from friends groups and also keep in view the website also.
Okay, sir.
Okay. Okay.
Sir,
Ah.
how can I get the PPT, sir?
PPT. Do you want PPT?
Yes, sir.
I will give you when you come to the practical training classes.